Hello students, welcome to the biology class. Today we are going to continue with our chapter biomolecules. In our previous session we discussed about the proteins, and today we are going to start with another macromolecule and very important macromolecule, that is your nucleic acid. What do you mean by nucleic acid? The term nucleic acid was coined by Altman in 1889 as it was known to occur only in the nucleus of cell. Nucleic acids are the polymer of the nucleotides. As we know, the adjacent nucleotides, they are held together by phosphodiester bond. A phosphate moiety links 3 carbon of one sugar of one nucleotide to the 5 carbon of the sugar of succeeding nucleotide. So with the polymerization of nucleotide, the polymerization of nucleotide will form the nucleic acid. Types of nucleic acid. We are having two types of nucleic acid. One is deoxyribonucleic acid, that is DNA. Another is ribonucleic acid, that's your RNA. Deoxyribonucleic acid or DNA that constitute the hereditary material for all the organisms except certain viruses, where the genetic material is RNA. In eukaryotic cell, it mainly occurs in the nucleus, and it is also being present in the mitochondria and chloroplast. DNA is the only molecule which is capable of replication, means that producing its own copies and producing a new DNA molecule of its own. So this is what known as a self-replication, producing its own types. Components of DNA. It is composed of 3 components: a molecule of pentose sugar called deoxyribose, a molecule of either purine, that is adenine and guanine, or a pyrimidine, that is your cytosine and thymine, and a molecule of phosphate. Now these 3 components, they are being present and they form the polymerization of nucleotide to form the DNA, that is your deoxyribonucleic acid. This is your deoxyribose sugar with the 5 carbon compound. At the carbon number 1, the base adenine is attached. Then there forms a phosphodiester bond at the 5 carbon position, and the linkage is from 3 carbon to 5 carbon. This is known as the phosphodiester linkage. This is known as a glycosidic linkage. So this is one strand of the deoxyribonucleotide, and there is the antipalar or the opposite strand of another deoxyribonucleotide. A means adenine, that forms a bond with thymine, and 2 hydrogen bonds are present in between A and T, and this is universally fixed that A will form a bond with T. Similarly, G means guanine, which forms a bond with cytosine with 3 hydrogen bonds. So one strand of the DNA runs from 5 prime to 3 prime, and another strands that runs from 3 to 5, they are antipalar to each other. Double helical, double stranded and antipalar. A will go with the T and G will go with the C. Now comes the molecular structure of DNA, that is known as Watson-Crick model. This is very, very important. So before we start with the salient feature, with the diagram we will understand the various features of a DNA. So as we have discussed, the DNA, it is having 2 strands. One runs from 5' to 3', another runs from 5' to 3' in the opposite direction, means they are antipalar to each other. They are having a deoxyribose sugar and they have a phosphate bond in between them. Sugar, phosphate, sugar, phosphate, sugar, phosphate. Now this sugar phosphate will form like this way. So they are having a sugar phosphate on this strand and they are helical to each other. So DNA is double helical structure and antipolar. Why antipolar? The one strand is from 5' to 3' and another strand is from 3' to 5'. And in between them, these stacks, they are equivalent to the nitrogen bases, that A with T with the 2 hydrogen bonds, G with C with 3 hydrogen bonds. So these stacks, they indicate that is the A with T and G with C with 2 and 3 hydrogen bond. This is complementary to each other. That is universally fixed, that A will complement with T and G will complement with C with 2 hydrogen bonds and with 3 hydrogen bonds. So that is a double helical structure. So they will make a turn. This is your one turn, and in one turn there are 10 nucleotide bases. Again, it is fixed in bDNA. So this is the major turn and this is your minor turn, and the one turn is of about 3.4 nanometer length, and the diameter or the width, you can say, that is 2 nanometers, and the distance between the one nucleotide is 0.34. So this is the dimensions of your double helical structure of DNA given by the Watson-Crick. Watson-Crick have proposed a double helical model of DNA. The salient features are: DNA molecule consists of two elongated polynucleotide chains of equal length. The polynucleotide chains are also termed as strands. Therefore, DNA is double-stranded molecule. This is not stand, this is strand. So be careful for that. The two strands are antipolar, that is run in the opposite direction, one from 5' to 3' and another from 3' to 5'. One end of the strand is called 3' end, whereas the fifth carbon of pentose sugar is free, and the other end is called 3' or 3', where the third carbon of pentose sugar is free. A spiral turn consists of 10 bases stacked at the interval of 0.34 nanometer or 3.4 angstrom. So they are having a 2 units. If it is nanometer, then it is 0.34, and if it is angstrom, then it is 3.4, because one pitch or one turn is 3.4 nanometer or 34 angstrom, maintaining the distance of 2 nanometer or 20 angstroms. The backbone of nucleic acid is made up of uniformly alternating pentose sugar and phosphate groups. The steps are made up of nitrogen bases, and hydrogen bonds hold the two strands together. Two hydrogen bonds are formed between adenine of one strand and thymine of another strand, that is A with 2 hydrogen bond with T, whereas 3 hydrogen bonds are formed between the guanine of one strand and cytosine of another strand, G with 3 hydrogen bond, that is with complement with C. This form of DNA is bDNA, which is right-handed and having a 10 nucleotides or 10 base pairs. So these are the very important salient features of model, that is a double-stranded model proposed by given by Watson-Crick. Now the second nucleic acid is ribonucleic acid, that is RNA. RNA is single-stranded polymer of ribonucleotides. So these are the polymers of ribonucleotides, that is adenine, guanine, cytosine and uracil instead of thymine in the DNA. Pentose sugar is ribose. The nitrogen bases are Purines, that is adenine and guanine, they are same. Pyrimidines, that is cytosine and uracil. Types of RNA, that is your ribonucleic acids. There are three types of RNA: messenger RNA, mRNA; transfer RNA, that is tRNA; ribosomal RNA, that is rRNA. The first is your messenger RNA. M should be small and then there is an RNA. So as the name is messenger, so it carries the information from DNA to ribosome in the form of codons. So these are the codons which are present in the form of a nitrogen basis, that is U is uracil, uracil, uracil and cytosine. It is equivalent to the particular amino acid, that is phenylalanine. So by this information, they carries the information from DNA to the ribosomes in the form of codons. So three nitrogen bases together, they form one codon and decide the sequence of amino acid in the polypeptide chain. So the basic function of RNA, they helps in the synthesis of protein while carrying the information from the DNA. Transferred RNA, that is your tRNA, means it carries a particular amino acid. So this is amino acid attachment from anywhere in the cytoplasm to the ribosomes, and there are as many tRNA as there are the amino acid. The structure of amino acid is like the clover leaves, so they are having various arms. This is called as an anticodon arm, that is with the tRNA. This is known as amino acid arm. So depending upon the anticodon, codon means again U, G, G. Along with that, they will form a amino acid over there. So here you can have a T loop, this is your D loop, and this is the variable. So this is the structure of your tRNA, which used to transfer the amino acid from cytoplasm to the ribosome, where the protein synthesis will takes place. Ribosomal RNA, that is, it forms a part of the ribosomes along with the proteins and thus forms a part of the seat of the protein synthesis. As we know, the ribosome is the organelle where the protein synthesis takes place with the help of RNA, that is called as your ribosomal RNA. So this is your messenger RNA, which is carrying the codons, and this is your ribosomal with the two subunits. It will get attached over there with the transfer RNA. So completely the protein synthesis will take place with the help of three RNA, mRNA carrying the message in the form of codon, ribosomal RNA providing the site where the protein synthesis can take place, and the transfer RNA or tRNA will carry the amino acid, so that the long chain of the amino acid will form, which is known as a polypeptide, which is the monomers of the proteins. So what is the difference between RNA and DNA? RNA is ribonucleic acid, DNA is deoxyribonucleic acid. RNA helps DNA to accomplish each task according to the traits and contains the genetic instruction used in development and the functioning of all the modern living organisms. This is single stranded, this is double stranded. Base pairing, adenine links to uracil, cytosine linked to guanine. It is adenine that links to thymine and cytosine links to guanine. Can be found in the cell's nucleus, its cytoplasm and its ribosome, found in the nucleus of the cell and in the mitochondria and the chloroplast. RNA is synthesized from DNA when needed, and DNA is self-replicating. The one more difference is in the RNA, we are having a ribose as the pentose sugar, and in the DNA, we are having deoxyribose as the pentose sugar. So with this, we finish our macromolecules, in which we discussed about the polysaccharides, we discussed about our proteins and DNA structures and RNA structure, that is in the heading of nucleic acid, and the model of DNA which was proposed by Watson and Crick is a very important model. So please go through it. And our next lesson will be on the enzyme of a same chapter. Till then, stay home, stay healthy. Thank you and have a nice day.